Hello, my name is Suman Rice, and I'm chair of the Society for Reproduction and Fertility. I'm delighted to welcome you to the SRF 75th Anniversary Lecture Series. This collection of talks celebrates 75 years of the Society, its rich history, and its ongoing contributions to reproductive science. The Society was founded in 1950 as the Society for the Study of Fertility, bringing together a remarkable mix of scientists and clinicians, from reproductive physiologists to endocrinologists, pioneers of livestock breeding, and experts in human fertility. Each lecture in this series highlights the diversity of research and innovation in our field, reflecting the interdisciplinary spirit that has defined the Society from the very beginning. We hope these talks give you a sense of the Society's achievements, the work of its members, and the dynamic community that continues to advance reproductive science.
Hello, I'm Tom Fleming from Southampton, and we're here to celebrate the SRF, the Society for Reproduction and Fertility, their 75th anniversary. So I would add that I think the SRF is probably the best learned society in reproductive biology. Make sure you're a member. Fantastic conferences, fantastic grants they give to particularly young people, and two great journals. So keep your membership up. And the idea for the 75th anniversary was that we'd take some of the key areas that are important in reproductive biology, an important remit for the SRF, and have a talk on each of those points, on each of those areas, to kind of identify what we've learned over these 75 years and give some updates, what are the highlights, etc. So my job here is to talk about the early embryo, and that's what I'm going to cover. So I think it's worth just stepping back for those of you not too familiar with the early embryo, the pre-implantation embryo, and just think about the huge number of activities that the embryo has to achieve in a very short space of time, like less than a week. It's a tiny structure and it has a lot to do. And I've just listed here how busy it is. It's got to sort out fertilization. It's got to incorporate the new embryonic genome and activate that genome. It's then got to reinitiate cell cycling. Remember, these have been dormant cells, the gametes, for a long time. They've got to reinitiate cell cycling. It's also got to think about metabolism. It's got to set up a metabolic capacity within this new embryonic organization. It's then got to go through cell differentiation. You'll see these pictures here of the early embryo. On the outside of the embryo will form the trophectoderm differentiation. And that major cell differentiation has to occur within a time frame. It's then got to segregate different lineages. It's got the inner cell mass, this cluster of cells that you can see down here. They have to separate from the trophectoderm. So lineage segregation or diversification is another job. It's then got to form a blastocyst, with the blastocele that you can see here. It's then got to think, right, I've got to hatch from the blastocyst, hatching from the zona pellucida, and then engaging in implantation. So this is a huge achievement for the early embryo, and it's tiny, and it's smaller than a full stop size on a printed page, much smaller. So I'm going to cover some key areas in this talk. So it's a bit historical, but I'm going to be very picky. I can't cover all of these points, so I'm just going to be very, very selective. So I'm covering these middle areas, how the trophectoderm differentiates, how the lineages of the inner cell mass and the trophectoderm segregate, and how you make a blastocyst, essentially, how it forms, and how you make a blastocele. So these are the more cell biological things. And I started working in the embryo area with Martin Johnson's lab in Cambridge many years ago in the 80s, and the cell biology was a key area that we studied then. And so from that track record, from that area, that time point, I'd like to cover this. Okay, so just starting with a very simple summary diagram. These are the three areas I'm going to cover: trophectoderm differentiation. I'm going to start with the mouse and where it's applicable, where we have the knowledge, because the mouse is where a lot of the mechanistic insights have been derived from. But I'm going to move also into the human to talk about what we know for the clinical embryology side. I'm going to concentrate on those two species. And so this is from the mouse. And you can see that for trophectoderm differentiation, shown in orange, it covers from the 8-cell stage through the morula and then into the blastocyst stage. It's still differentiating on the outside of the embryo, the TE, as we call it. And then lineage segregation happens at the 8 to 16, as the 8 cells divide to form the 16-cell embryo. Then allocation of cells to the interior of the embryo can be seen in blue here. This is the inner cell mass. And similarly, further cells are allocated at the 16 to 32 cell stage. And then subsequently, the inner cell mass segregates into what remains as blue here, the epiblast, which goes on to form the fetus. And the primitive endoderm, shown here in yellow, which goes on to form the yolk sac placenta. And I should say that the trophectoderm itself goes on to form the main chorioallantoic placenta after implantation. And it's the trophectoderm that interacts with the endometrial cells of the uterus to engage in implantation. So we're going to cover those areas, and I'm going to start with trophectoderm differentiation. And as I say, highlight some of the key discoveries over the time of the SRF. So let's just look at compaction first. So yeah, let me put that in context. Here is when the embryo undergoes a great change. The cells become adhesive, closely opposed to each other in a process called compaction. And we see that diagram in here, and we can see this in the photograph here. The cells, the blastomeres, are clearly identifiable here, and then you can't see them individually as compaction kicks in. It's achieved through E-cadherin cell-cell adhesion, shown here by an immunofluorescent. But at the same time, the cells polarize from the outside as an apical surface deep to the inner part where it's the basal surface. So all cells polarize and they form an apical pole of microvilli. So you can see this down in the scanning EM photos down here. These are from Martin Johnson's lab back in Cambridge days. And here embryos where the zona pellucida has been removed. And you can see here before compaction, you have this image where you can see all the blastomeres. When compaction has occurred, you can see how the cells have clustered together very tightly. And if you remove calcium, because this E-cadherin is calcium dependent, you can see the pole of microvilli very nicely on these blastomeres here. And that's shown here on this transmission EM photograph. And at the same time as the microvilli polarizing, you also get polarization inside the cell. Many organelles, like these endosomes, cluster in what we call the apical surface, the outward-facing surface of the cells. And also the cytoskeleton polarizes with actin and microtubules. We'll come to some of that in a moment. So how does compaction occur? Well, there was a lot of studies considering this issue. How does that E-cadherin adhesion system kick in? And studies have shown that all the protein components of the junction, the E-cadherin junction called the adherens junction, summarized here in this picture, E-cadherin as a transmembrane protein and a cluster of different catenin proteins which link the cadherin to the actin cytoskeleton. Well, it was shown that all these proteins were being expressed by the early embryo, But then particular studies from David Kapko's lab in Arizona shown that it was a post-translational mechanism that activated compaction. And he identified phosphorylation from protein kinase C of the beta-catenin protein, shown here in blue, that became phosphorylated. And that allowed the whole junction to adhere together, became functional, became active, and the embryo compacted. So a post-translational mechanism there. And also to flag the work from particularly Rolf Kemmler's lab in Germany, where during the 80s and 90s, knockouts of specific genes involved in this process were achieved, and knocking out E. cadherin itself stopped compaction, stopped embryo development, and it was a lethal condition. So those were two important discoveries in that. So one more recent paper that really interesting for compaction has been one from Fierro Gonzalez in Nature Cell Biology in 2013 from the lab of Nicholas Plachter in Monash, where he showed by injecting RNA, fluorescent RNA or E. cadherin GFP RNA into one blastomere, it would express E. cadherin, and you'll be able to see it in live cell imaging. And you see these fantastic filopodia spreading out from one cell to the next and actively pulling one blastomere to compact with its neighbors, as shown here in these pictures. And this was a really important discovery, I think, and shows the dynamic nature of this interaction between the blastomeres. Okay, so these filopodia. Yeah, and if you induced them early, you could induce compaction early. So they're very important in a functional sense. Or if you blocked the filopodia from forming, you could block compaction. So further studies, particularly from Magdalena Zernicke-Gertz's group in Cambridge, were able to establish much more detail on the polarization process, because this polarization is the first step in the differentiation of the epithelial polarized phenotype of the trophectoderm. And they were able to show that many different proteins were present in this apical domain, ESTRA in part 6, protein kinase C, zeta in particular. And this apical domain became very important in organizing the remainder of the cell polarity. It organized the cytoplasm and organized the junctional sites. And you would also at the same time get the first formation at the border between the apical domain and the basolateral domain, an actomyosin ring, and the formation of the first tight junction proteins that would eventually be important in morphogenesis of the embryo, which I'll come to very shortly. Okay, so this apical domain was seen as a primary regulator, and we'll come to that again, and the importance of that shortly. So further work from Magdalena's group in Cambridge showed that that polarity and the organization of the cytoskeleton within these 8-cell blastomeres also include localizing specific messenger RNAs to specific regions of the cell. And one key discovery was the CDX2 messenger RNA, which is critical in specifying the trophectoderm fate. Was localized in the apical cytoplasm, as we can see with this in situ staining. And that maintains that message within the outer trophectoderm lineage by stabilizing to the cytoskeleton and to utilize in transcription to generate the trophectoderm epithelium. Looking back at the human, we see the same process of compaction in human embryos, but they occur slightly later, more towards the 16-cell stage in many cases. And in many embryos, you get full compaction of the embryo, all the blastomeres engage in compaction. But in some, as you can see in the bottom here, cells can be excluded in the human embryo. And when you look at the clinical potential of those embryos, they are slower to develop, they have a poorer blastocyst potential, and ultimately, if they're transferred, they lead to a lower live birth rate. And Giovanni Coticchio from Bologna has some very nice studies in this area. The human embryo also shows that same organization of the apical domain, again with the in the microvillus pole, part 6, PKC zeta. So very similar cell biology between the two species. So it's important. Now, trophectodome differentiation will continue, but I'm holding here just to bring in how do you generate these two lineages, the inner cell mass from the trophectodome, from the 8-cell stage. And this is done by a process of differentiative division, asymmetric division, which I'll show in the next slide. So lineage segregation. Each of these blastomeres at the 8-cell stage, which is polarized, when they come to divide, aspects of their polarity are retained, and when the cells go through division, they can do from across the apical basal path, across the apical basis axis, to generate a polar daughter cell and a non-polar cell shown here in blue that enters into the embryo interior. Other cells divide conservatively along the axis of polarity to generate two polar daughters. And so this generates a pool of cells on the inside of the embryo. And this same process occurs at the 16-cell stage when division occurs to the 32-cell stage. And when this occurs, you get a variable number of these divisions in the 8-cell embryo, but it's inversely related to the number that are generated in the 16-cell stage. So more asymmetric divisions in the first round will mean usually less in the second round, and vice versa. And that's work from Martin Johnson's group, particularly, that's looked at that. More recent developments included this very nice paper showing that the spindle poles, when they form for these divisions, in many cases, the pole of the spindle can be captured by the apical pole, and as a consequence, induce an asymmetric division. So a further mechanism whereby the apical pole can interact with the spindle to organize a polarized, an asymmetric division and generate cells to the interior of the embryo. Another mechanism has been the importance of actomyosin cortical tension between the blastomeres that are forming at the 16-cell stage. A number of them, there is a strong cell-cell adhesion that can actively draw cells to the interior of the embryo, as well as these asymmetric divisions. So there's a contribution from the actin cytoskeleton to maintain these two lineages and to separate them. A further way in which this apical pole can retain and maintain the distinct cell populations that are generating has been shown by what's called hipposignaling. And this reinforces the lineage segregation. So what happens in the outer cells that are differentiating to form trophectoderm, the apical domain captures and inactivates protein kinases AMOT and LAT1-2, which allows a transcriptional coactivator, YAP, to enter the nucleus and promote expression of CDX2 and other trophectoderm transcription factors to maintain the differentiation of trophectoderm. Whereas in the inside cells, where there's no apical domain, these kinases, AMOT and LAT1-2, can phosphorylate YAP and block it by phosphorylation from entering the nucleus, and therefore no longer induce the expression of CDX2, but instead can express through transcription factors factors that will promote inner cell mass fate. So again, we see the importance of cell contact patterns and the apical domain in generating and maintaining these lineage diversifications. And so lastly, just want to talk about morphogenesis. And I should say that these processes in Hippo are present in both the mouse and the human embryos. So again, very important for clinical research, the understanding of these mechanisms. So as I said, lastly, just to talk about the process of morphogenesis. In the mouse, we see that the blastocyst forms at the 32 cell stage, okay? Why is it then? Why not later? Why not earlier? So there's a timing mechanism here, which we think is very important, which then allows for the blastocoele to form and the formation of a full blastocyst. And this is where tight junctions become important. Some of our own work in this area. So we go back through these images of the embryo going through cleavage. From the time of compaction, as I mentioned before, proteins that are important in generating the tight junction, which acts as a seal holding cells together and allows them to then transport fluid across the epithelium to generate the blastocoele. These proteins form at this apicolateral contact point, and it's gradual, it's sequential. Some proteins are expressed and assembled there at the 8 cell stage, some at the 16. You can see more of these proteins accumulating at this junctional point. But then at the 32 cell stage, the final step is achieved, and the proteins can function and actively seal the tight junction, and the blastocoele cavity can then form. And what we think is the key regulatory event is the late expression of a single isoform of the Z01 protein called Z01 alpha plus, because this has the function of binding other transmembrane proteins in the cytoplasm at the Golgi. You can see these interactions here, and brings it to the cell surface, brings it to the contact point, and allows then the junction to seal. So specific transcriptional activity of a splice variant we think is very critical in the timing of blastocyst formation. And if we look at the human embryo in this context, you see the same pattern of gene expression and protein assembly at the junctional complex, that same sequential pattern that we saw for the mouse. But very interestingly, we often see in the human embryos failure to express the Z01 alpha plus, that important critical regulator, as you can see in these pictures here. And what can happen then is tight junction formation can be incomplete. The membrane proteins present inside the cytoplasm are, it appears, unable to reach the junctional site. And as a consequence, these embryos have been compromised and often result with a lower score. And you can see that blocked assembly in these pictures here. So I've given you an overview, a rather simple overview, of some of the key events that happened in mouse and in the human, and similar events happen in other species. I'm just going to finish with a couple of additional points. One is this question: does the mammalian embryo engage in what's been called pre-patterning? Is there some organization in the zygote that can specify the processes that I've been talking about, the generation of the trophectoderm epithelium, the formation of the inner cell mass, and the formation of the fetus? And some of the work, particularly from Magdalena Zernicke-Gertz's group, and this has been demonstrated in both mouse and human. If, for example, you differentially label 2-cell blastomeres, they will divide at different times to continue their development. And early dividing 2 cells, she was able to show, then subsequently engage in more asymmetric divisions at the 8 and 16 cell stage, and thereby generate more cells within the inner cell mass. and thereby generate a larger proportion of cells in the developing fetus subsequently. So patterning and organization way back at the start of cleavage from the zygote could be very important in the developmental process for both mouse and for human. And this would, in some ways, link to lower organisms which do engage asymmetries within the zygote itself to generate lineages during development. But this is not necessarily deterministic. It's more a guide to processes that may subsequently occur that we've just been discussing. It's still a controversial point, but it's been one of the big developments in recent years. Very much to the present day now, the question particularly for fully understanding human development and the ethical issues of not being able to work extensively with human embryos has developed into the use of stem cell lines from trophectoderm, from the epiblast, and from the primitive endoderm, which cell lines that can reassemble in very specific organizations that can become what's known as a synthetic embryo or an embryoid. And these processes, this self-assembly of these different cell lines has allowed, through the use of important engineered platforms such as microfluidics and micropatterning in microwells, able to identify much more clearly the signaling, the cell interactions, the differentiation events that might be occurring in normal embryos. And in that way, we can study beyond up to the gastrulation stage for some of these processes. And that's a major advance and will have important clinical implications down the line. And the last thing to mention on the human clinical embryology is the importance of AI in clinical art success. One of the major developments that we've seen in human embryology has been the use of time-lapse microscopy in the incubator, where without disturbing the embryo, you can build up from video images a morphokinetic pattern, including embryo cleavage, compaction, and other dynamics. That information generating algorithms that can help select the best embryo. And from these algorithms, a massive information now, from those algorithms, the question of which is the best embryo to transfer becomes one that can be studied. So can deep learning select the best embryo for transfer? And this is in recent randomized controlled trials, compared that with an embryologist using the more traditional view of conventional morphology for selection of the appropriate embryo. And now randomized double-blind controlled studies have been conducted to see which is best. Can the AI or the embryologist, who wins the day? And it seems that there's no significant difference. Here we see in this study 46% correction for a clinical pregnancy with deep learning, whereas 48% with an embryologist. So there's still work there for an embryologist rather than just directly based on AI algorithms. But the AI is quicker, more efficient, and is improving all the time. So this is just a snapshot in time. There are very interesting studies there on the embryo. And lastly, I've just talked about the kind of internal processes of how a blastocyst is made, some of the key mechanisms that have been identified over the years. But that's only half the story. The other half is the embryo is also looking outwards, checking its environment and adapting how it develops, dependent on the nutritional supply that it might be receiving. And we found that many diseases in the adulthood can be programmed from the very early embryo environment. So it's not just a question of making it an embryo by inward mechanisms, but it's also a question of the embryo looking outwards and to select the right pathway to survival. Lastly, I want to thank, just from my career, lots of funding bodies and lots of people that have worked in my lab. And thanks to a lot of friends and collaborators over the year. So long live the embryo and long live the SRF. Thank you very much. And I'll stop there. By the way, if you need to email me about any aspects of what I've talked about, please feel free. There's my email address, and then do that. Thank you very much.